Welcome back to our series of lectures. Today we are talking about the second golden age of microbiology. That is part 4 of the history of microbiology. This is our lecture 16. The lecture is presented by Microbiology, the biological sciences. Kindly feel free to contact us through our contact listed below for any comments or assistance. Thank you. This is the period when medical microbiologists focused on the search for substances that could destroy pathogenic microorganisms without damaging the infected animals or human. The treatment of diseases by using chemical substances is called chemotherapy. Chemotherapy is also commonly referred to chemical treatment of non-infectious diseases such as cancer. Chemicals produced naturally by bacteria and fungi that act against other microorganisms are called antibiotics. Chemotherapeutic agents prepared from chemicals in the laboratory are called synthetic drugs. The success of chemotherapy is based on the fact that some chemicals are more poisonous to microorganisms than to the host infected by the microorganisms. The first synthetic drugs. Paul Ehrlich was an imaginative thinker who gave the first shot in the chemotherapy revolution. As a medical student, Ehrlich speculated about a magical bullet that could hunt down and destroy a pathogen without harming the infected host. In 1910, after testing hundreds of substances, he found a chemotherapeutic agent called salvarsan, an arsenic derivative effective against syphilis. The agent was named salvarsan because it was considered to offer salvation for syphilis and it contained arsenic. Before this discovery, the only known chemical in Europe's medical arsenal was an extract from the bark of a South American tree, quinine, which had been used by Spanish conquistadors to treat malaria. By the late 1930s, researchers had developed several other synthetic drugs that could destroy microorganisms. Most of these drugs were derivatives of dyes. This was arrived at because the dyes synthesized and manufactured for fabrics were routinely tested for antimicrobial qualities by microbiologists looking for a magic bullet. In addition, sulfonides, the sulfur drugs, were synthesized at about the same time. The first antibiotic was discovered by accident. Alexander Fleming, a Scottish physician and bacteriologist, almost tossed out some culture plates that had been contaminated by molds. Fortunately, he noticed the curious pattern of growth on the plates, a clear area where bacterial growth had been inhibited and circulated by molds. Fleming was looking at a mold that would inhibit growth of a bacterium. The mold became known as Penicillium chrysogenum, and the mold's active inhibitor was called penicillin. Thus, penicillin is an antibiotic produced by a fungus. The second golden age of microbiology began in the 1940s when the enormous usefulness of penicillin became apparent and the drug came into common use. Since these early discoveries, thousands of antibiotics have been discovered. However, use of antibiotics and other chemotherapeutic drugs have some problems. Many antimicrobial chemicals kill pathogenic microorganisms but also damage the infected host cells. Toxicity to humans is a problem in the development of drugs for treating viral diseases. Viral growth depends on life processes of normal host cells. Thus, there are very few successful antiviral drugs because a drug that would interfere with viral reproduction will also likely affect uninfected cells of the body. And thus, over the years, you have a challenge in development of the organisms have developed resistance to antibiotics that were once very effective against them. Drug resistance results from genetic changes in microorganisms that enable them to tolerate a certain amount of antibiotic that would normally inhibit them. For example, a microorganism might produce enzymes that inactivate antibiotics, or it may undergo changes to its surface that prevent an antibiotic from attacking to it or entering it. The recent appearance of vancomycin-resistant Staphylococcus aureus and Enterococcus faecalis has threatened health care professionals because it indicates that some previously treatable bacterial infections may soon be impossible to treat with antibiotics. The quest to solve drug resistance, identify viruses, and develop vaccines requires sophisticated research techniques and correlated studies that were never dreamed of in the days of coach and pasture. Other microbiologists also use these techniques to investigate industrial applications and roles of microorganisms in the environment. Bacteriology. This is the study of bacteria. It began with Van Leeuwenhoek's first examination of tooth scrapings. Many bacteriologists, like Pasteur, looked at the role of bacteria in food and environment. In 1997, Heide Schultz discovered Theomargarita namibiensis, a bacterium large enough to be seen with the unaided eye. This bacteria lives in mud on the African coast. The bacterium consumes hydrogen sulfide, which is toxic to mud-dwelling animals, and this makes it a unique bacteria because of the size and the inhabitant. Martinus Beijerinx lived between 1851 and 1931. A trained botanist, formulated enriched culture technique. For instance, he devised a precise chemical defined medium to isolate rhizobia and proved that they were responsible for the formation of the root nodules in legumes. Using the same technique, he isolated the first pure cultures of many soil and aquatic microorganisms. Sergei Vygotsky lived between 1856 and 1953, studied bacterial diversity of soil and water, particularly bacteria that recycle nitrogen and sulfur compounds. Sergei Vygotsky showed that Bagiatoa, commonly observed in marine sediments, catalyzed specific chemical transformations in nature and proposed the concept of chemolithotrophy, that is, oxidation of inorganic compounds to yield energy. He showed that they are widespread in nature and obtain their carbon from carbon dioxide. Using enriched medium that lacked nitrogen, he isolated anaerobic nitrogen-fixing bacteria, Clostridium pastorinium, and became the first to demonstrate the process of nitrogen fixation. Mycology. This is the study of fungi. It includes medical, agricultural, and ecologically important branches of fungus. Fungal infections rates have been rising in the past decades, accounting for about 10% of hospital-acquired infections. Climatic and environmental changes, that's like severe drought, are thought to account for the tenfold increase in Coccidioides immitis infections in California. New techniques for diagnosis and treatment of fungal infections are being investigated. Parasitology. This is the study of protozoa and parasitic worms, largely known as the helminths. Because many parasitic worms are large enough to be seen with the unaided eye, they have been known for thousands of years. It has been speculated that the medical symbol, the rod of Asclepius, represents the removal of parasitic guinea worm, that is, Dramancalus medinensis. Asclepius was a Greek physician who practiced about 1200s before Christ and was identified as the god of medicine. At the sideways is the image of the symbol of medicine, immunology, that is commonly the study used of the immunity and the immune system. Knowledge about the immune system has accumulated steadily and expanded rapidly. Vaccines are now available for numerous diseases including measles, rubella, mumps, chickenpox, pneumonia, tetanus, tuberculosis, influenza, whooping cough, polio, hepatitis B, malaria, and even typhoid. The smallpox vaccine was so effective that the disease has been eliminated. Also, the polio virus is estimated to be eliminated as very soon. In 1933, Rebecca Lansfield proposed that Staphylococcus be classified according to serotypes based on certain components in the cell wall of the bacteria. Streptococci are responsible for a variety of diseases such as sore throat, streptococcal toxic shock, and septicemia. In 1960, interferon substances generated by the body's own immune system were discovered. Interferons inhibit replication of viruses and have triggered considerable research related to the treatment of viral diseases and cancer. Virology is the study of viruses. It originated in 1892 when Dmitris Levanovsky reported that the organism that caused mosaic disease of tobacco was so small that it passed through filters fine enough to stop all known bacteria. At the time, Levanovsky was not aware that the organism was a virus. In 1931, Wendell Stanley demonstrated that the organism, tobacco mosaic virus, that's TMV, was fundamentally different from other microorganisms and so simple and homogeneous that it could be crystallized like facilitated the study of viral structure and chemistry. After the development of the electron microscope in the 1930s, microbiologists have been able to observe the structure of viruses in details. Martinez generics use selective filters to show that the infectious agents of tobacco mosaic disease, that's virus, was smaller than a bacterium and that it became incorporated in the cell of the living host plant. Thus, he described the first virus and basic molecular genetics. Virology. Microbial genetics is the study of the mechanisms by which microorganisms inherit traits, and molecular biology is the study of how genetic information is carried in molecules of DNA. In the 1940s, George W. Biddle and Edward L. Tatum demonstrated the relationship between genes and enzymes. DNA was established as the hereditary material by Oswald Avery, Colin MacLeod, and Maclean McCarthy. Joshua Lederberg and Edward L. Latham discovered that genetic material could be transferred from one bacterium to another by a process called conjugation. In the 1950s, James Watson and Francis Crick proposed a model for the structure and replication of DNA. In early 1960s, Francis Jacob and Jacques Monod discovered messenger RNA, that's ribonucleic acid, a chemical involved in protein synthesis, and later they made the first major discovery about the regulation of gene functions in bacteria. In the same period, scientists were able to break the genetic code and thus understood understand how the information for protein synthesis in messenger
Although
RNA is transmitted
genetics
into the
encompasses all organisms,
amino acid sequence
much
for
of
making
knowledge
proteins.
of how genes determine specific traits has been revealed through experiments with bacteria. Unicellular organisms, primarily bacteria, have several advantages for genetic and biochemical research. Bacteria are less complex than plants and animals, and the life cycle of many bacteria lasts less than an hour, so scientists can cultivate very large numbers of bacteria for study in a relatively short time.